Hi everyone, I'm Julie Karam and I'm a summer scholar in the Summer Research Training Program in Biomedical Big Data Science at the MION Lab. Today I'll be presenting on the project that I've been working on, which is GeneBot and DrugBot, a pair of Slack applications to discover knowledge about genes and drugs. To begin, I'll be giving some general background on Slack. This application is a tool used for group collaboration that compiles the communication styles of email, text, and instant messaging into one app. Slack is an acronym for Searchable Log of All Conversation and Knowledge, so one of the principal goals of the app is to not only centralize and streamline, but also store everyday communication between teams. Over the past couple months amid the COVID-19 pandemic, the demand for remote working has increased exponentially, and consequently, Slack has experienced remarkable user growth. This is illustrated through the figure here on the slide, which displays the trend in created work teams from January 3rd to March 23rd. As you can see from the pink line, there was a clear spike in the number of newly created teams after March 12th, when many companies were forced to start working remotely. Compared to the projected number of new teams based on the previous 2 years, it is evident that the company has been greatly influenced by the pandemic. So there are countless reasons why companies may decide to utilize Slack, but one of the largest appeals of this platform is the ability to customize the workspace. Slack has their own app directory that has over 2,200 Slack applications, as well as an API that allows users to easily create their own Slack apps. And while there are teams from all different fields that utilize Slack, biomedical research groups have become some of the most prolific users of this platform. But despite this fact, there are essentially no Slack applications geared towards this field currently available. While there are countless apps that can ease workflow for those in finance or programming, there still remains a great opportunity in developing biomedical-focused Slack applications. As a computational biology lab, there are countless tools that we utilize every day. Some to get information about genes or drugs, such as Gene and NCBI database, or DrugBank, one of the largest drug databases. Others are tools that our lab has created to help perform different analyses, such as Enricher or ArchES4. If someone decides to research a gene, for example, they might visit various databases, and when they decide to share the information with others, they'll have to go through the process of compiling all these sources. The simple task of researching a gene turns into a tolling one. But let's say there was an application that did all this for you, from gathering a summary to attaching figures and links, right to Slack by simply calling a command. Well, from this idea is where GeneBot and DrugBot were born. GeneBot and DrugBot were our solution to bridge the gap in biomedical app integration into the online workspace. They're a pair of Slack applications that provide information about genes and drugs utilizing bioinformatics databases and analysis tools for the Slack workspace. These apps integrate the search and compilation of information and data analysis, easing the share of information and leading to streamlined communication within teams. Now I'll be presenting the various functions of these applications. The first function that GeneBot offers is the ability to retrieve a summary about your gene of interest, as well as gather links to gene landing pages from various databases. Both of these applications were designed to trigger and perform different functions through the implementation of / commands, which are a feature on Slack that are essentially shortcuts that allow users to quickly trigger a certain function for a certain application. In this case, the /gene command will output a gene summary. The way this works is that after the command is called, the text of the message will be retrieved from the event data sent from Slack to the application. This text, which should be the gene name, is queried using the Gene API. If a gene is found, the bot will retrieve the summary for that gene from the API, as well as generate links to harmonosome, ArchES4, and GeneShot, and then post this all to Slack. For example, if a user types /gene2, the summary will be displayed as well as links to various tools, making it very convenient for the user to explore more information about the gene of interest, as well as share all the information with teammates. Next, I'll be going over a very similar function, but in DrugBot. The /drug command provides a drug summary and information about the user input. The way this works is that after the command is called, the text of the message will be retrieved from the event, and this text, which should be the drug name, is queried through DrugBank and L1000 metadata. since drugs can be identified by different names. Then the bot will retrieve a link to the database depending on which ones it's found in. As you can see here, if the user inputs /drug methotrexate, the following will be displayed. In addition to providing information about genes and drugs, GeneBot and DrugBot both allow users to submit a list of genes for enrichment analysis right in Slack. In GeneBot, this is achieved through the /geneset command. Users can either type their gene list or upload a CSV file of their list of genes. Users can also specify which library they want to be displayed in Slack by typing the enricher library name or shorthand name, which can be found in the app's user manual. The gene set is then submitted for analysis with Enricher using the Enricher API. GeneBot will retrieve a link to the geneset specific landing page for Enricher and display this along with a bar graph that shows the top 10 enriched terms for the selected geneset library based on p-value ranking. As you can see here, there are various input formats that GeneBot will recognize, and in this case, if the user put that input, then the bot will recognize the first term as the library and the rest as the genes. And then the data outputted from Enricher is used to generate the bar graph, which shows a visual representation of how significant each term is based on the overlap with the user's input list. And in addition, the bars are colored based on significance as well, with the darker colors representing a more significant term. In DrugBot, there is a function that works almost identical, except this time users will submit a drug list for enrichment analysis, and the drug enricher API will be used to analyze this data. Here's an example where a user uploaded a file with their list of drugs, and DrugBot outputted the link and then information about it as well as the bar graph. Another function that GeneBot offers is the ability to perform gene network analysis based on protein-protein interactions and gene-gene co-expression data for a user-inputted gene list right in Slack through the /network command. Similar to the enrichment analysis function, users will input their gene list after calling the function. The genes will then be analyzed using the Genes to Networks API to receive results of a protein-protein interaction subnetwork that connects the enriched genes and proteins with known protein-protein interactions. The genes are also analyzed through a human gene correlation matrix from ArchES4, which provides the pairwise Pearson correlation of genes across expression samples. GeneBot will retrieve the top 5 correlated genes for each inputted one. And combining these two sets of data, GeneBot then generates a network graph to display all these gene-gene interactions. This figure, as well as information, will be displayed in Slack. As you can see here, if a user types /network BRCA1, BRCA2, STAT1, MAPK3, GeneBot displays the gene set and number of genes inputted, as well as this network graph. As you can see from the key, the red nodes represent the seed or inputted genes, while gray nodes represent the intermediates. The co-expression relationships are shown by the pink edges, while protein-protein interactions are shown in blue. In addition to being able to get information and perform different analyses, we also wanted GeneBot to serve as an educational tool. So we created the /learn function to promote the knowledge of genes to help keep scientists and researchers aware and informed about their field of study. When the command is called, GeneBot will output an interactive multiple choice question with 4 answer choices where users can match the correct summary to the gene in question. GeneBot randomly selects 4 genes from a curated list and uses the summaries of these genes as the answer choices. From this set, 1 gene is randomly selected for the question, and users can then choose their answer, and GeneBot will respond whether they're correct or not. As you can see here, when a user types /learn, a question will be outputted. In this case, what is ESR1 or estrogen receptor 1? If the incorrect or correct answer is selected, GeneBot will respond accordingly. Those are all the functions that are currently available as part of GeneBot and DrugBot, and these applications should be available for download in the Slack app directory. Overall, GeneBot and DrugBot provide a novel approach to genomic data analysis and biomedical research in the context of Slack workspaces. In terms of future directions for the applications, I plan to implement more functions, perhaps a drug synergy function for DrugBot to help better understand drug interactions for diseases, or adding additional visualizations for the enrichment analysis functions. I'd also like to improve the /learn function to create an even more interactive Slack game with features such as time limits, multiple players, and scorekeeping. Also, I think it'd be really cool to implement natural language processing methods to improve the text processing for the bot. This way, the bot can begin to learn which text refers to genes and drugs rather than always parsing through inputs, and this would create an even more streamlined user experience as the bot would start to be able to predict what the user wants. Lastly, I would like to thank all the lab members and summer scholars for all the continued support and inspiration throughout the summer. It has been such a remarkable experience working with everyone, and I've been able to learn so much over the past 10 weeks. Special thanks to Avi for his wonderful guidance and mentorship, and Sherry for being so kind and organizing the program so well, and my collaborators Alone, Errol, and Max for helping me every step of the way and teaching me so much, and Nicole for designing the wonderful bot logos. Thank you.